What's up, guys? Welcome back today. Today I'll be talking about biology C1.1, enzymes and metabolism. It will both have the SO and HO content. I'm pretty excited for today's video. Sorry if it's like the last day before exam, but yeah, I've got everything you should know about this unit. So let's get started. First of all, enzymes as catalysts. So what are catalysts? So what's important is that a lot of reaction we need our body needs a lot of reactions, right? But they happen too slow naturally, and it's not fast enough to support like our life function. So catalysts speed up chemical reactions, and in organism these specific biological catalysts are called enzymes. So what they do is, you see this grandpa, he's a very slow reaction, but it makes it into a very fast reaction. So the reaction will still get there at the end, but it just makes the reaction occur faster, and enzymes are not used up, so they are reusable. So yeah, makes a fast reaction. And what is metabolism? So metabolism is what we call every single chemical reaction that occurs in our organism, and it's controlled by a specific enzyme. Okay, so it's really important that every single reaction has their own enzyme and only that enzyme. And so that reversibly means that this enzyme can only control this reaction. So because we have so many reactions, it's good to know that there are so many enzymes in every organism, including humans. And how I like to think of it is my phone is a iPhone SE. So it's an old one. So it can only use the old charger, not the new charger. So think of it, this reaction can only be charged by this enzyme. So it's a one-on-one thing. And in a chemical reaction, there's the reactants and the products, and the enzyme like helps the process go faster from the reactants to the products. But this chemical reaction will occur anyways, but enzymes are only making it faster. So just saying again, important to know. So what is anabolism and catabolism? So anabolism builds macromolecules from monomers. So it's building the big thing from a lot of small things, and it requires energy. And in a lot of scenarios, it'll have condensation, which means it'll release water through binding. And some examples of it are photosynthesis, protein synthesis, and forming glycogens. Now, catabolism is basically a complete opposite. It's breaking down macromolecules into monomers. So breaking a big thing to a small thing, and it can release energy, and it uses up water through hydrolysis. And some examples include oxidation and digestion. And both of these are the two different types of metabolism. There's chemical reactions, and so there's like building up and also breaking down. And that brings us to energy and life. So our organism needs a lot of different kind of energy, and then the kind is heat, and we measure in kilocalories. Okay, it's mentioned in the textbook. Good to know, kilocalories is 1,000 calories, and 1 calorie is the amount of energy needed to heat up 1 gram of water or 1 ml up 1 Celsius. Okay, and the energy currency in of a cell in our bodies is called adenosine triphosphate, or ATP, and it's what our cells use most. So here are some four like pretty common energies, and let's see if you guys know what it means. So this is what a kinetic energy is. It's the energy of movement. Potential energy is a stored energy that can be used. It's got some potential. Chemical energy is the energy, a potential energy, that was stored through a chemical reaction. So this energy will be released when a chemical reaction occurs. And thermal energy is, the heat, the temperature. So it's a form of kinetic energy through the random movement of particles. So what's ATP? Adenosine triphosphate. It's not APT, although it's pretty similar. So ATP is, you can see the structure here in a photo. It has a adenine, a ribosome, and it's connected to those three phosphates. That's hence the tri. So triphosphate, and through hydrolysis, it releases one phosphate and unleashes energy. Not unleashes, releases. And it becomes ADP. So it has two phosphates. It releases a phosphate 3 to 2, hence the diphosphate, or ADP. It's pretty good to know these structures. And through phosphorylation, adding the phosphorus back, that's where you add energy into it to put it back to ATP. So ATP is really useful. It supplies energy for almost everything in our bodies, including anabolic reactions. If you guys remember, it's building small from big. You need energy for that. For mechanical work, running or like moving the flagella, I guess. And active transport, when you're having active pumps from like low gradients to high gradients. You all use ADP, so important energy. Globular proteins. So almost all of enzymes are made of proteins, and enzymes are long amino acid. Remember, amino acid is the monomer for protein, and it's in a globular shape. So a globular shape over here is a 3D structure, and it's very specific. And why is this specific? Because I mentioned before, each enzyme only works for one particular reaction, okay? And we'll see why that's important. A substrate is the thing that the enzyme will be working on. It's the specific object that adheres to the enzyme. So the active site is the place where it matches up. So, uh, oh, sorry, I thought I put in a wrong slide there, but it's all right. So the active site is only composed of a few amino acids, but the function of the active site is it goes back and forth with the entire 3D structure of the globular protein. So the entire 3D shape is really essential for the enzyme to work. And if the shape of the entire enzyme is moved permanently, then the enzyme will be denatured. And denatured means that it's not folded anymore and it can no longer be a catalyst. And denaturalization happens due to a lot of different factors, which we'll look at later. So yeah, the active site is really important, and it's where the substrate binds to the enzyme. And the new thing here is the induced fit model. So before we were taught to learn about the lock and key model, but that's wrong. Ha! They lied to us. Well, they didn't. They just simplified it. But we know better now. So both the substrate and enzyme change shape to allow for a tighter fit. So how I like to think of it is you have a glove, and a glove is like to hold on something. So in order to hold onto it, like a balloon, for example, you need to change the shape of your hand, the glove, to hold the balloon, and the balloon will also change shape. So both the enzyme and the substrate will change shape when it's binding. And this is possible because the protein molecules in making the enzymes, the amino acid long chains, are flexible. And enzymes, what enzymes do is they weaken the bonds within the molecules of the substrate in order to break them apart. So enzymes and the substrate, it doesn't always fit perfectly, but it has to be the right shape still. There's only one correct shape. There's only one correct substrate for every enzyme. And the active site changes shape slightly to hold on to the substrate better. So that's the induced fit model. Now, activation energy. I don't know why, but we skipped directly from 1.1 to 0.4 to 1.1 to 10. That's cool. We'll skip that later. That's how the textbook writes it. So, substrates require kinetic energy for the reaction to occur. And there's two ways to overcome this. So, first, we can increase the kinetic energy through temperature. But in a lot of organisms, you don't want to be like too hot. That's bad. So, in a lot of organisms, we use a second way. Use enzymes to lower the activation energy. And the reaction will occur until there's an equilibrium between a product and a reactant. So activation energy, I think of it like Sisyphus, he's pushing rock up the hill. You'll get a reference later in this slide. So the activation energy is the energy necessary to destabilize existing bonds in a substrate. You want to work on it. So you need some energy to like get it going. And enzymes lower the activation energy required for the reaction. So if you look at the graph here, we can see like with an enzyme, the hill that the rock needs to be pushed up is much lower. It still needs activation energy and still needs some kinetic energy, but it's much less steep, so it's easier to happen. And when it's easier to happen, it happens more often because not every time they have as much activation energy as it requires. And these enzymes, they decrease the activation energy and they're not used up and they can be reused. Okay, so exergonic reactions happen when they're creating bonds. And endergonic reactions happen when you're breaking bonds. So you're either releasing a lot of energy or you're using up a lot of energy through this activation energy. Molecular motion. So when you want the enzyme to bind with a substrate, you don't just need enough activation energy. That's the movement. You also need the correct orientation. You want it to fit. You need to put the key in the lock the correct way. Although it's not a key and lock model, but you still need to put it in the right way. So that's the orientation. And some immobilized enzymes are the substrate or enzyme that's embedded in a membrane to make it more efficient. And some benefits of this is that it can be reused easily. It's easy to purify and it exposes the enzymes better to the substrate or vice versa. And a common application of this in real life is making lactose free milk. So lactase is the enzyme that breaks down lactose for those lactose intolerant people. So um, people use these beads that have lactase on them. They're immobilized. They're stuck on the surface of beads, and they pour milk through it in order to break down the lactose, and that will create lactose free milk. So that's an application of using the immobilized enzymes, and it has some benefits to it. And here are the 6 or 7 steps of the mechanisms of enzyme action, and it's basically a summary of what we just learned before, but I think it's pretty useful to know all these steps. I won't be reading everything out, but it's just good to know. This is in a textbook. Enzyme substrate complex, it's the thing that happens when they bond. It's temporary, like they're together, they're still reacting. And the takeaway from this subunit is that the structure of the enzyme is really important, guys. The shape is really important in this situation. And now we'll look at the effects of some different things on enzyme on action, or the rate of reaction. So temperature's effect on enzymes. So when temperature increases, that's more kinetic energy, right? So that's more activation energy. And also more collision because molecules move faster when there's a higher temperature. So the rate of reaction will increase as the temperature goes up until it reaches the optimum temperature. However, if the temperature is too high, then enzyme will get denatured. And when I just said that enzyme gets denatured, I mean that the shape is changed. And you don't want the shape to be changed because that means the enzyme just doesn't work anymore. So when the temperature gets too high, it's denatured and the rate of reaction goes down really rapidly because you're basically breaking all the enzymes and they don't work anymore. You're breaking the machines. It's like you're flying too close to the sun, but then like you get burned. The pH value. So there's an optimum pH for every enzyme, and it's can be neutral, it can be acidic, it can be basic, and it's suited to the environment. So the closer to the pH, the more optimal it is for the enzyme, and the faster the rate of reaction. So for example, pepsin is the enzyme in our stomach, and we know that the stomach is really acidic, so its optimal pH is at 2, which is acidic. For trypsin, which is an enzyme in our intestines are slightly basic, slightly alkaline, so its optimal enzyme pH level is a bit alkaline. And what that means is that um, you want your enzymes to be at the correct pH, so not too low or not too high, and it'll both decrease the effectiveness of the enzyme. Lastly, substrate concentration. So the substrate is the thing these enzymes are working with, right? And if you have more enzymes, then they can keep working. And the more substrate you have, the more they can work at once. However, if you're adding so much substrate that all of the enzymes are already working, then adding more substrate won't help, right? If you're in a factory and you have a lot of machines, but all the machines are already working, it won't help importing goods inside. So this is the graph that will occur using a relation of the rate of reaction to substrate concentration. So you can see it will gradually reach a ceiling as all the active sites are occupied. But before that, the increase in substrate concentration will increase the rate of reaction because it will increase the opportunities for collisions and for these enzymes to occur reactions with the substrate. 1.1.9, measuring enzyme reactions. Basically, you want to measure enzyme reactions the exact same way. You want to measure other rates of reactions, and it's to do with some math. So for this one, I think it's pretty straightforward, but if you like, you can do some practice on Cognity about like identifying the graphs maybe, or doing some very easy algebra math. But this is pretty straightforward if you like pass like pre-algebra, I think. But back to the content. HL content. So SLs, bye bye. But still watch if you want to learn, new things. Maybe it'll come in useful and you can become a smarter person. Um, so there are intracellular and extracellular reactions, both for enzymes. So intracellular in a cell, and enzymes are going to be made by the free ribosomes in the cytoplasm. And some examples are the Krebs cycle that happen in the mitochondria in the matrix, and glycosis, which occurs in the cytoplasm. Now, extracellular is not in cell, it's outside the cell, and it's made by bound ribosomes. Where is it bound? It's bound to the ER, the rough ER, endoplasmic reticular. Yeah, I think I said that right. And some examples include chemical digestion in the gut. So these enzymes are released out of the cells and they occur out of the cell. So that's extracellular reactions. A term here I want to bring up is multi-enzyme complexes, and I'll come back later, where it's like it's a series of different reactions needed to get to a product. So it's a lot of steps together, and every single step needs its own enzyme. So that's why it's called a multi-enzyme complex. Heat generation. No reaction is perfectly 100% efficient, and there will always be some energy lost. And in most often ways, it's lost as heat. And scientists think we only use about 35% of the energy we have because we lose all the rest, which is sad, but I guess it's enough. And for endotherms or warm blooded animals such as the duck and us, the heat is used to maintain the body temperature. So whenever there's an enzyme reaction, it'll release heat. Whenever there's a metabolic reaction, it'll release heat. It's losing the energy, but I guess it's okay. Metabolic pathways, there's two. There's linear and there's circular. A linear is a line. This is a multi-enzyme complex that I said before. You need a lot of different reactions to get to a specific goal you want. And some examples of a linear is glycolysis. I'll show one of it later. You don't need to know everything about it. And an example of circular is the Krebs cycle or the Calvin cycle. And this is the glycolysis, and you can see it's not exactly linear. It's a bit more complicated. It has branches. It has so many different enzymes, so many reactions. But linear reactions, you can know it's a linear reaction because the end product isn't the same as the start product. However, in a circular reaction, you can see it's a circle, and the end product will be like the starting product here, where you start off with citrate and you'll go back to citrate in the Krebs cycle. You don't need to know this, but I think this looks so scientific, and it'll be really cool if I can like say it out of my heart. And the Krebs cycle is gonna come up again. Forgot where, but it will come back. Inhibition. What is inhibition? Inhibition decreases the rate, the productivity, the efficiency of these enzymes. And how does it do it? There's a lot of types. There's competitive and non-competitive. So what competitive enzyme is, it competes. It competes with the substrate to bind to the active site. So it has a similar shape to the substrate. So it tricks the enzyme in. I mean, it doesn't trick. It doesn't have a brain, but it goes into it has the same shape, so it can fit into the active site, the induced fit model, like slightly. And because it's already in, the correct substrate can't get in. And this inhibition, this competitive inhibition, doesn't work. And most of the time it's reversible, but sometimes it's irreversible. And an example the textbook gave is statins. So statins is for people that have cholesterol problems. And what statin does is it competitively inhibits the reaction that occurs to like create cholesterol for those people. It's a drug that doctors give. And this is impacted by substrate concentration because the more substrate you have, um, the less chance that these competitive inhibitors can go fit in the active site before the substrates, right? And for non-competitive, let me check the time. Non-competitive efficient inhibition, this will bind to another site, not the active site. It's gonna be the allosteric site, and it can be reversible or irreversible. Most of the time, it's reversible. For example, there is metallic ions and also cyanide. So cyanide is a really common poison, and it's really deadly, man. So it's permanently disable your functions and you die immediately. Scary. And this is the graph that happens for non-competitive inhibitors and competitive inhibitors when you increase the substrate for the rate of reaction. And feedback inhibition. So your body wants to be efficient, right? So when you have enough of the end product, you don't want to create any more, you don't want to waste any more. So the end product sometimes it inhibits the first reaction. So it stops this process of making it, and when all the end product is used up, there's no more inhibitors, so this process keeps going on. And the example that we need to know is isoleucine. So it's amino acid that our body needs. And last thing, mechanism-based inhibition is also called suicide inhibition. That's pretty tough. And the example we need to know is penicillin, where it binds to transpeptides irreversibly. So transpeptides are the things that make up the cell walls of bacteria. And you can remember penicillin, it was the first antibiotic, and it saved a lot of lives because it can kill the bacteria by binding to the transpeptides, and the bacterials will die because their membranes burst. And bacteria can mutate to become resistant to penicillin by changing the structure of the transpeptides in their cell membrane. So yeah, that's all the content in C1.1 on enzymes and metabolism. I hope you enjoyed today's video. I really enjoyed making today's video. And remember to subscribe. And good luck. This is Alexander Fleming. He's the one who invented penicillin, discovered penicillin, and I hope everybody gets a 7 tomorrow. So, yep. Subscribe, and I'll see you in the next one. Bye bye.